very much, all of you, for joining the investor event this evening. I'm hoping that there will be quite a number of new investors who do not know about Angle or do not know very much about Angle. So the idea of this presentation is to give you some background on what we've been doing in Angle and why we believe that we're an investment that's certainly worthy of your attention, and particularly attention at this point in time. So Angle is a highly innovative UK listed company, and we are pioneering a new approach to cancer care, which is called liquid biopsy, and I'll explain what that means in this presentation. And we believe that our liquid biopsy solution, which is involved in recovering intact cancer cells from patient blood, has the ability to transform cancer care for all types of cancer. So both the early diagnosis and monitoring of cancer, and also therapy selection to guide treatment. Now, we have put a lot of effort into developing a very clever system for isolating and then harvesting for analysis cancer cells from patient blood. Now, why would cancer cells be in patient blood? Let's say the patient has breast cancer or lung cancer or prostate cancer. Why would they have cancer cells in their blood? Well, the reason is that that's the way that the cancer spreads. And in fact, over 90% of patients who die from their cancer eventually have died from the secondary cancer that develops, not from the primary. So in the case of breast cancer, and it equally well applies to all solid tumor cancers, but I'll give breast cancer as an example, the patient's cancer may spread to other organs, notably in breast cancer, that's the bones and the brain and the lungs and the liver. And it's that spreading of the cancer which can cause the ultimate progression and demise of the patient in the sad cases where that happens. So the way that the cancer jumps from the breast to these other places is via the blood circulation system primarily. And the cancer cells that leave the primary and go into the blood and then are spreading the disease are called circulating tumor cells. And these are intact living cancer cells in the blood, which are essentially moving from one location to another. It's been very well understood that these cancer cells are present in the blood. The problem is getting hold of those cancer cells because they're very rare in number. There might be one cancer cell in a billion blood cells. So that could translate, for example, to a 10 milliliter tube of blood containing 10 billion blood cells and maybe 10 cancer cells. Might be 20 cancer cells. If the cancer is progressing very fast, it might be 200. There might be no cancer cells if the therapy that the patient is on is working nicely, or if the patient is in remission. So the challenge is, how do you get these cancer cells out? Now, the other important bit of background is the biology of cancer, which is that each patient's cancer is different from another patient. So if you have 3 patients with breast cancer, you imagine they have the same disease, but in reality, they have a disease in the same part of their body, but it may be different between patients, and therefore the standard of care is to give the patient a tissue biopsy. So cut out some of the cancer, take it to a laboratory, examine the cancer cells that are present in that sample from the tissue biopsy, and from that make the decision as to which drug is appropriate for that patient, how extensive should their treatment be, etc. So the care pathway is determined based on the status of the cancer cells in the tissue biopsy, and this is the same across multiple cancer types. The challenge, however, is not only is it different between patients, but it also changes over time, and that means that the information that the doctors are using to treat the patient and make decisions on which drugs are appropriate becomes out of date, and they actually end up effectively guessing. What they'd like to do is repeat the tissue biopsy, but obviously by that time the patient's had surgery, so they might have had a mastectomy or a lumpectomy, and there isn't an obvious location to go and collect more cancer cells to repeat the biopsy. So doctors are left with the old information to guide their treatment decisions. So what we're pioneering is a new approach. Take a simple blood test, recover the cancer cells from that blood sample, and then they can be examined to work out how the cancer is evolving and therefore what would be the most appropriate treatment decisions for that individual patient. So our solution is shown on the slide here as an instrument, the Parsortix system, which is a machine which automates the processing of the blood, and a consumable, which is like the razor blade in the razor blade model. So it's a one-time use Parsortix cassette, which is a patented microfluidic design that captures the cancer cells based on their somewhat larger size, and importantly also on their lack of deformability. They compress not nearly as much as blood cells, which can squeeze through little tiny gaps. So we flow the blood sample on an automated basis through the Parsortix cassette. The plasma liquid and the blood cell component, the white blood cells and the red blood cells, can flow straight the way through the microfluidic cassette, whereas the cancer cells are retained in the Parsortix cassette and thereby separated from the blood components. The machine automates that. Once it knows that that process is completed, it will automatically wash away the remnants of the blood cells, and then it will reverse flow and recover the cancer cells. So then you have a pristine, living, intact cancer cell recovered from the patient's blood, giving you real-time information on what's happening in the cancer's evolution. And we've been through a process to get an FDA clearance, which is the Food and Drug Administration in the United States. We're the only company in the world that has achieved that for recovering intact cancer cells, harvesting for subsequent analysis. That was a 6.5-year process of investment and very significant work by the company. What we now have embarked on is developing a number of different methodologies for analyzing those cancer cells. And what we're effectively doing is taking the established techniques which are used for analyzing cancer cells from tissue biopsy and redeploying them and refining them so that they can work on a small number of cancer cells that come from the patient blood. And that, we believe, provides the best possible sample for repeat biopsying of patient condition to guide treatment going forward. And also it can be used for the early detection of cancer in patients who may be asymptomatic. It's a minimally invasive process because all it requires is a blood test for the patient, and it's effective, it's very reproducible. It's also affordable and enables repeat testing. So we've addressed a critical issue in the care for cancer patients, which is how do you get up-to-date information on what's happening in their cancer. And obviously the status of cancer is actually increasing significantly, the incidence levels, both demographically and across different age groups. So cancer incidence is stated by Cancer Research UK to be as high as 1 in 2. So 50% of all of us on this call are likely to be touched by cancer. The treatments are becoming more and more expensive. So the new treatments can cost £150,000 per patient, and yet they only work for perhaps 1 in 5 patients. So the cost of treatment is soaring, and therefore the process of liquid biopsy that we're pioneering can both improve the cancer patient outcome, in other words, make sure they get good treatment, which gives them a better outcome, but also has a major role to play in controlling healthcare costs, which are spiraling out of control at the moment. By enabling the use of the technology to select which drug will be successful for which patient, you can reduce the wasted spend, which is about 80% of drug expenditure at present. Now, we are driving forward the commercialization of the Parsortix system by leveraging relationships we're building with pharmaceutical companies. We're doing this in order to benefit from the funding that these large pharma bring to the whole development process. So we secured our first large pharma contract early this year, which was with Eisai, the large Japanese pharmaceutical company. Revenue's a bit over nearly 4 billion, 10,000 employees. They are actively developing a new class of drug called a HER2 antibody drug conjugate. They're not the only people doing this, but it's a very important drug class because the antibody drug conjugate targets the cancer cell and only unloads its payload when it's attached to cancer cells, or at least that's the aim of the antibody-drug conjugate. The antibody binds on the cancer cell, and in this case, because it's a HER2 antibody in breast cancer, it will bind onto HER2 positive breast cancer cells and then unload the drug. The purpose of this is to make it much more specific. So essentially, traditional cancer therapies like chemotherapy will cause a lot of side effects and toxicity. That's why patients' hair will fall out and they'll have digestive problems and other problems. It's because the drug is attacking healthy parts of the patient, not just the cancer, because it's going for fast-growing cells. So the antibody-drug conjugate class of drugs is targeting the actual proteins on the cancer cells before unloading the drug payload. So that raises a question as to whether or not the patient is in fact got HER2 positive cancer. About 1 in 5 patients will have HER2 positive or HER2 low, about 1 in 5 will have HER2 positive breast cancer, and then possibly another 20% will have HER2 low, and they can be targets for the antibody drug conjugate. But the critical point is that these drugs are prescribed as the cancer develops generally, and the HER2 status changes over time. So it's already known that over 40% of breast cancer patients, their HER2 status changes. So Eisai needs to know what is the current HER2 status of the breast cancer patient. So the reason Angle is a solution for them is because we offer the ability to get intact cancer cells from a patient blood sample, and we've now developed a HER2 assay to analyze those cells for their HER2 status. So Eisai has engaged with Angle with a view that we will ultimately provide them with a companion diagnostic for their approved drug, which would mean that every breast cancer patient would have multiple blood tests using the Parsortix system in order to determine whether or not their HER2 status is positive or HER2 low, in which case they could be given the Eisai drug. So this is an opportunity for us because Eisai is paying for, or will be paying for, all of the clinical trials and all the development work that's necessary to bring this drug to market. In fact, they've already committed a $2 billion investment program for this drug, and we'll be a relatively smaller part of that, but we can earn a lot of money from the clinical trials as they pay to use Parsortix in their trials. And we've developed a very focused clinical laboratory within Angle to provide these services. So the patient that goes into the Eisai drug trial has blood drawn from them by the hospital, it's shipped to Angle, and we have all the processes to run that blood through Parsortix, recover cancer cells, and then look at their HER2 status. So that's the first one. The second one we announced later this year is with AstraZeneca. Very sort of similar idea. They're looking at measuring a protein on the cancer cells that are recovered by the Parsortix system. In this case, they're interested in the DNA damage response proteins, and we've developed a method for measuring those. You can't look at these proteins any other way than getting circulating tumor cells. So competing liquid biopsy technologies of circulating tumor DNA, which is fragments of dead cells, don't work for these proteins. There's a very large-scale investment program within AstraZeneca focusing on DNA damage response as a new class of drugs. And what this is to do with is preventing the DNA damage repair that the cancer cells go through. So the idea is you have a combination drug, a chemotherapy to attack the cancer cells, and then a DNA damage response inhibitor to stop the cancer cells repairing themselves. And again, to do that, you need to understand which cancer cell proteins are shown on the cancer cell, and are they actually a target for that particular drug. And again, the good thing here is that this is fully funded by AstraZeneca. They're paying us to run initially some development work, but the aim is that we'll get in multiple phase 2 trials with AstraZeneca in the DNA damage response area. And of course, there are multiple other pharmaceutical companies interested in the similar things, both the antibody drug conjugates I mentioned earlier and the DNA damage response. There's a big conference going on in Boston at the moment where we have colleagues attending, which is focused in on companion diagnostics in Boston at the moment. There's a number of other companies talking to us about this kind of thing. We've got a third contract with large pharma this year already, which is again with AstraZeneca. And in this case, they're focusing in on prostate cancer. Again, they need us to measure a specific protein called androgen receptor. In this case, we didn't have the ability to do this just yet, and AstraZeneca are paying us just shy of £600,000 to develop the ability to look at androgen receptor in prostate cancer cells. So again, this is a third major set of contracts which we've developed. All of these three that I've just described and others like them that we're seeking to secure with other pharmaceuticals and also with the existing ones can go through a process where there's an initial phase of a pilot study or a development phase, and then we can get into phase 2 trials, which could generate revenues for Angle between £1 and £4 million each. There are multiple opportunities relating to one with Eisai, and then multiple relating to the AstraZeneca contracts, DDR and androgen receptor. And then once the company's successful in the phase 2 trial, you can get through to phase 3, and they can be up to 1,000 to 3,000 patients in each trial, which means that we could earn between 15 and 45 million pounds for each one of those phase 3 trials. So this is a really innovative commercial approach adopted by Angle to align ourselves with some of the largest pharmaceutical companies in the world, to give them something that they cannot otherwise get hold of, and basically give them the ability to assess how well their drugs are working and to develop companion diagnostics. So that business area for Angle is going extremely well. We've got an integrated business model, so we are selling with distributors, we're selling the Parsortix instrument and the Parsortix consumables with global distributors. That gives us the opportunity to combine that with our services model. So when the customer finally gets to companion diagnostic, they need to have a solution that can be rolled out to hospitals worldwide, and that's what we offer with our product side of our business. So the services and the products come together extremely nicely to give us a combined solution. In terms of why the pharma should do this, I'm going to wrap up very shortly so that we can have maximum time for questions, but there's some big advantages for the pharma in aligning themselves with Angle. Notably, they can increase their trial success. So clinical trials have only about a 1 in 8 chance of getting all the way through, and they cost hundreds of millions of pounds. If they select the patients by looking at the biomarkers that they can get with the blood test from Parsortix, they can significantly increase their clinical trials probability of success. They can also fail quicker, which sounds a bit obtuse, but actually failing quicker means that they can cut their investment costs as quickly as possible and save money on those trials that wouldn't get all the way through. They can also increase the efficacy of their drugs. By selecting the right drugs for the right patients, they can get much higher response rates from patients, which means also has a knock-on that they can get a regulatory clearance more easily, and a knock-on that they can charge a lot more money for the drugs. The higher the success rate and impact on positive outcomes for patients, the higher the amount of money that the pharma companies can actually charge for their drugs when they come to market. And obviously, on top of that, they're in competition with multiple other pharmas. Remember that the top drugs can earn anything between $1 billion a year right the way through to $20 or $30 billion a year for their owners. So there's a massive prize for these companies. But obviously, there's multiple other companies competing with them. So the better they can select the patients that should be given their drug, the better the outcomes and the better their pricing and the better their competitive position. And the area that we're looking at, analyzing intact cancer cells, is something that they can't do with other suppliers in this marketplace. We've got a series of other smaller pharma customers as listed on this slide. The slide deck will be available after the presentation. One thing that's absolutely critical in our space of medical diagnostics is the third-party information that's available to demonstrate performance of these, of whatever the medical diagnostic is. And a big side benefit of the fact that we sell our equipment as a product is that we now have third-party independent cancer centers utilizing our equipment. And in fact, there are now 41 leading cancer centers around the world who have undertaken research studies and other investigative work, which has been published in 94 peer-reviewed publications in world-leading journals, which are peer-reviewed. So, and it works without modification in 24 different cancer types. So this is my final slide, and then please bring Ian in to join the questions. We've got, following our fundraise in June of this year, we're now in a strong position to deliver commercial progress. We've got multiple areas of development. The pharma services side, I said, is really beginning to develop well. We've got 3 large pharmaceutical, large pharma contracts that are being developed all the way through the process I showed, and we're working on securing others. Our products have been developed. We've added content, so different solutions for analysis of the cells onto our ability to recover those cells. So we give an end-to-end solution. And we're also running multiple clinical studies in progress. And notably this year, we have published some initial data on analyzing circulating tumor cell DNA compared to ctDNA, and we found that we can identify clonal evolution of the cancer in a way that cannot otherwise be done. So that's a summary presentation. I'd like to take questions now.
So first up, what size of blood sample is needed, and is the angle technology different from Grail?
I can answer that. We have the ability to be flexible, so we actually have a small volume adapter, so we can actually work with mouse models, which is very important in research aims. The standardized tube is a 10 milliliter tube, and that's sufficient for all of the downstream analysis. I think the second part was, how does it differ? So the main difference is that the bulk of the industry is focused on analyzing circulating tumor DNA, which is fragments of dead cells, which is quite a limited analyte.
Just to add that, you mentioned Grail specifically. So Grail is focused on multi-cancer early detection. So it's basically geared towards a blood test for asymptomatic individuals. And if you look at the underlying results that have been published to date, they've done quite well in a few cancers, but they're quite poor in a number of other cancers, particularly some of the major cancers such as prostate and breast cancer. So obviously Grail is sort of, everybody likes that, but it has many challenges on whether it'll be adopted. Obviously there's a big trial being run by the NHS. They declined to adopt it early, but I do think that there's some merits in it. But I think it's more going to be geared towards symptomatic patients rather than asymptomatic, because the economics are otherwise not really cost effective.
Okay. Next question. Does the company hold patents on its products and processes? How do you protect your intellectual property to prevent other companies benefiting from the technologies developed without Angle's approval?
Yeah, Ian, do you want to answer?
Yeah, so we've got patents out to 2035. There's multiple families, so we've got good protection there. But quite rightly, that's only one component. As Andrew mentioned earlier, we've got the first and only FDA clearance. That took a long time to achieve and a lot of money, so that's a significant barrier to entry. We're second most published system, so we've got a good sort of first mover advantage there in terms of other systems. The one system that's been around a lot longer only gets epithelial cells, and that has many limitations, and that's partly why CTCs were less adopted in the industry, because of the limitations of this legacy system. We've overcome those limitations. The cells we capture, you can do all forms of analysis on them, and that's what is really is changing the mindsets, that this was a technology or an analyte, the CTCs, that didn't work. We're now proving we can capture them consistently, repeatedly, reliably, and therefore they can be used in assays. And hence we haven't sort of changed the minds of the market, but we are achieving that, and we're beginning to see those contracts come through.
A couple of questions about the existing pilots. They're both kind of asking the same thing about the SI pilot and the Astra. When would you expect to know if you'll be involved in follow-up trials?
So we've got the all three of those contracts are now progressing very nicely, and one would expect that by the end of this year, beginning of next year, we'll have completed the first phases of those, and then as soon as they're ready, we'll move into hopefully phase 2 or and then subsequent phase 3 trials. And as I was seeking to mention, the ESI one is very targeted on antibody drug conjugate and HER2. There are many other people interested in that, so there's quite a halo effect. We've got other conversations going, and the ones with AstraZeneca are both of high interest to others too. There's many DDR companies, and androgen receptor is a standard for prostate cancer. I think there's 135 clinical trials running focused on androgen receptor in prostate cancer right at the moment, so there's quite a wide open opportunity.
Okay, I think time for one final one. And I mentioned we had questions come in before the webinar, so I'm going to make sure we get through one of those. And with UK funding proving to be hard to source as ever, would you look at a US listing in the future? And would that be at the expense of AIM, or would you have a dual listing, or how has it been something that's been discussed?
Ian, do you want to cover that?
Yes, I mean, we've certainly, if you look at the depth and breadth and the pools for capitals in the US, then the US is an attractive market as you scale the company, but you've got to get the timing right. There's plenty of companies that have gone there too early, and it's very, very expensive to list and operate there. AIM's been very good for us. To date, we've always managed to raise the necessary funds and get the support from institutions, and we already have our two largest shareholders, both US, so US investors can invest in us as an AIM company. I think it's the balance of getting the timing right. We are sort of lined up, our auditors are PwC, we've got a December, I know they sound straightforward things, but they're all things you need to do for NASDAQ listing. We do have an ADR as well, but you've got to be careful about going to the US too early. The reason I mentioned that it's a possibility is that, as Andrew highlighted with our technology, it does work with every cancer it's been tried for, and it does work across every part of the patient pathway. Therefore, there's a huge opportunity in front of us, and to properly exploit that, you would need a substantial amount of funds. But at the moment, we're following a much more tightly constrained model with the amount of the cash and resources available to us.
Okay, Andrew, Ian, both thank you very much indeed for joining us this evening.